is Dr. Annie Couturier, and welcome to our podcast, Conversations with an Organ Engineer. On today's episode, we'll discuss what artificial intelligence is and its role in tissue engineering.
Hello everyone, welcome back to our channel, and I'm Kai, and this is Dr. Couturier. And today we are actually going to dive deep into an exciting intersection of science and technology, that how data science or artificial intelligence are transforming organoids research. And if you don't know anything about organoids, check our past episodes, and we'll give you more. We are giving you more information about organoids in the next few episodes. And before we talking about actually diving deep into AI intelligence, so we're gonna talk about what type of data can we derive from the organoids, and what actually that data analysis will related to our daily healthcare, or what kind of information it contains.
Yeah, so Kai, with our organoids who haven't seen our previous episodes, we encourage you back, as Kai said, to go look at our previous episode. But organ or organ-like structures, we call them organoids. We use induced pluripotent stem cells to generate various kinds of organ-like structures in the human body, consisting brain, liver, kidney, intestines, and so on and so forth. But what can we do once we've generated them in the lab? What's the purpose of this? To generate some kind of analysis, and that's where data comes in. And the way we analyze these organ or organ-like structures is we have different kinds of way of analyzing to generate data. So before we get into artificial intelligence and train machines to analyze data, we need first to know what kind of data we produce. So we use, first of all, we can cross-section them, which just means to cut the organoid into different sections and then look at it under a microscope. This way we can look at the cellular and molecular morphology of the cells. So what do the cells look like? What's their shape? What they are functioning as. We can look at all of that. We can do it in live tissue, so we can do this in live microscopy. We can do it at a fixed point using immunohistochemistry, again, which just means fixing the tissue at a certain point and then cross-sectioning, so cutting it out and then looking at cells. So we could do it at both time points. We can fix them, we can do them live, we can track their development. And that's one particular data set. Then the other thing is we can literally extract their RNA, DNA, and we can look at the gene expression data. So we can extract genes, or we can extract RNA, DNA from these tissues and look at what genes are being expressed, what time period they're getting expressed at, what is their physiological relevance to the pictures that we've taken. And we can do all of that using gene expression, and we call in our scientific terms transcriptomic data. So that's one. And then, so that's the second type of analysis. So we did imaging, we did transcriptomics, which is gene expression data. The last one the third one we look at mostly is protein data. So similar to the gene expression data, we can also extract proteins from this tissue and look at proteins. So what proteins are being expressed at that point, how are they being expressed, how much are they being expressed, can all be assessed as well. So that's another set of data. So we've gotten to 3 now. The fourth set of data, depending on the tissue you have got, we want to do some functional output. How do you know a tissue is behaving like itself, like it does in the human body? We use functional analysis, and we call it functional data. So for example, the best one I can give is brain tissue. Usually our brain cells talk to each other using electrical impulses, and we call them synapses, and those are chemical in nature but generate electricity, and they talk to each other using this electrical current. And so we can measure that current. So that's called electrophysiology. And we use multi-electrode array systems where we have electrodes that we have, and then we place a little organoid on top of it, or brain structure on it, and then we can measure the electrical activity of the brain, and then say that, yeah, this is a functional brain because it is producing electrical activity. That means the neurons are talking to each other. So that's another set of data. Now, this can be done for cardiac cells as well, or cardiac organoids. Cardiac, as we know, produce electrical activity as well, and they beat. So we can measure their electrical activity and their contractility, so how much they contract. And again, we use multi-electrode array systems, which are electrodes to measure both things. For example, another kind of tissue that we have used is lung tissue or intestinal tissue, where they form these tight junctions, and there are very, very thick tissue that we can measure how thick they are. What is the conductance across their thickness of the tissue? And that's another way of measuring the tissue integrity for a lung or intestinal organoid, and we call that impedance. So there's various ways of measuring functional output. We can also measure, now that's like when you go to your doctor, you go and say, oh, can you measure when your tummy's off or when your liver is not functioning properly? They do a liver function test, and they have all these enzymes they can measure. And we can do the same. We can do the same in the tissue that we produce and see all the enzymes that are being produced by our liver organoid. Is it producing bile? Is it producing liver enzymes that you can measure? And you can measure that in the tissue itself and the media. Media is the supplement that we provide to keep these structures alive. So we have a plethora of data that we get from this. And this is where your machine learning and AI comes in. We generate a lot of these organoids, and we generate them in good quantity. And then, as I said, there's like this 6-7 formats of data analysis that we perform. We need to make sense of them, and so we can combine all of this data. And that's where your machine learning and AI comes in.
I see. So another thing is organoids now are widely used in disease modeling. So and since we're talking about data science and artificial intelligence, can we actually utilize artificial intelligence to cultivate some of the biomarkers for disease diagnosis, or that contribute to even personalized therapeutics? Since last time when we talked about, one of the advantage of organoids is that it is derived from patient-specific iPSC, which will carry over patients' genes, like from themselves.
Yeah, that's a great question. So first we need to define what a biomarker is in terms of an organ structure. So yes, you're absolutely right. We can do that. And there are various scientists working in this field. So one of the things that we do is using our, let's say for an example, a brain organoid, and we derive it from a patient that is schizophrenic. So we work mostly in mental health. So, and going back to our personalized medicine episode as well, we talked a lot about mental health related diseases and how organoids are a great way of measuring or looking at these cells. So what we can do is, a biomarker in an organoid is the cellular features and it produces that we can measure using, again, microscopy and proteomics and genomics. So we can measure the cellular biology of it, and that's their cellular signature. So we can call that as a phenotype or a cellular biomarker. And how do we correlate that back to the patient is we have been doing this in multiple ways. For example, it is widely known in schizophrenia that they produce less inhibitory enzymes, and they're called GABA. So the inhibitory enzyme is commonly lower or not found as much as in a healthy person. Schizophrenia shows GABA to be lower. And we have seen that in our protein data as well as gene expression data for schizophrenia-derived organoids. So this is an example of how we can correlate patient data to organoid data. It not only validates our organ engineering as a model, but it also gives us a step further. So now I have a patient population, as you pointed out, a personalized medicine approach. Go back to our episode to just check out that out, FYI. But for that GABAergic cohort of patients, let's say there are 10 patients of schizophrenia, 5 of them show that they are producing less GABA in their blood tests, in their brain tissue, whichever way they were assessed, but they're showing less inhibitory enzymes in their blood test or CSF panels, so cerebrospinal fluid panels. And if they're showing that, and we derive organoids from those patients, and instead of giving them, oh, let's try this drug or that drug or this drug, there's a plethora of drugs again. So we try the drug on the organoid that is also producing from the same patient less GABA and say, okay, there's 10 drugs. This is the 10 drugs I'm going to try on the organoid, give a combination of those. And then if that works on the organoid, it's a very high likelihood it will work back in the patients. And this is where AI comes in. It can gather patient data, it can gather organoid data, it can mesh it, and it can tell us if that this is the drug you need to give. So not every time you even have to make the organ or organ engineering it, because if we have hundreds and hundreds of these patient samples, we can ask the machine to learn and recognize that this patient with this symptoms is going to behave like this, their organ is going to behave like this, so give them this medication. But that is not yet in practice. That is years away, and I'm hoping scientists like us can make it a reality.
Okay. And so since we talk about like artificial intelligence that can be integrated into diagnosis, and we just want to know like how good we are right now, and like could you like give us kind of like imagined like scenario what could be a paradigm in the future that by integrating artificial intelligence in like healthcare or illness diagnosis, like what will be like the life when you're walking into like urgent care?
Oh, that's a very good question. It is interesting for neurological and neuro-related disorders, because I'm a neuroscientist by training. It will become a world more easier. I think within the next 10 years, we should expect someone who has or is experiencing neurological symptoms, comes in, talks to literally a robot. I'm expecting it to either talk to like those vending machine kind of paradigm. And this is my opinion, and it can be something different altogether. And says, this is my symptom, this is what I'm experiencing, and then it itself goes into hundreds and thousands of patient symptoms that it has recorded and says, okay, yes, you are type A of this kind of disease, and these are the tested medications, but it won't tell the patient. It should still let the doctor decide, but it will, the time it takes to get to the doctor to decide and all of that, that clinical support decision will be already be done and analyzed and already there. So it took you a couple of months to get to the neurologist. The neurologist had to assess you, do the test, do this, do that. That will get skipped over. And with AI and organ and tissue engineering, we expect is where organ engineering would come in this is like your tissue needs repair, or you had a stroke and you went to the urgent care, you were diagnosed immediately with a stroke. We would have some stem cell therapy to replace the cells that were damaged immediately. And that can be done either by the neurosurgeon on the team or a robotic neurosurgeon that could help it. That may be in 25 years. But this is right now science fiction. Right now, I think we're at the stage where we're realizing how important AI can be and how we need to responsibly integrate it into medical care.
I see. That's fascinating. Like, I hope someone like one day can become true.
Yeah. I'm hoping to push the needle forward where we can use these stem cell or stem cell related products to bioprint or print organs that can be used for transplant. That's our goal in our lab. We're already working on one project such as this where we can transplant organoids, heart-related organoids into animal tissue, animals, for showing that you don't even need a transplant. You had a heart problem, you come with that heart problem to us, we make the tissue from your cells and transplant it back to you. That would be the goal for, and where AI can come in is analyze the patient, what tissue they need, what problems they're going through it, and they can give us that information, and it takes us so long to get to that data that half the time, and by that time we can make that organ that you need.
Mm-hmm. And last, I want to talk about, so basically right now, one of the hot topics, everyone's talking about it, is the AI tools like large language model, like ChatGPTs. Everyone is using it, but that also raise a lot of concern about data privacy and security, because with organoids, since it's derived from the patient, and which will contain a lot of patient sensitive data, so a lot of people will concerned about their data privacy, and how good are we, or what kind of effort we have already made to protect those privacy, or would that be one of the problem we will be concerned in the future?
Yes, it is a very, very good problem to be concerned about, and there have already been steps taken. So any patient cell line or patient sample that we get, it's de-identified. So, which just means, for example, I donated my cells to make stem cells. The only information that a scientist would get in our lab for organ engineering particularly would be their age, sex, and their diagnosis and what medications they were on, just because we need to know how they react to this medication. But that also can be taken off. We will not, right now this is already in place. We have to fill up forms. We are very diligent about it. The review boards, international review boards, the stem cell regulatory body is already in place. So with us organ engineers, the tissue and the stem cell lines are very, very safe. We do not have any information that is relevant to the patient. But now the next step is the clinicians will have all the information, and they're also governed by the same regulatory bodies. So if they use artificial intelligence in their systems to analyze the data that we provide to them, it is up to the hospital and the medical administration to provide the data security. It's going to be treated the same way. This data should be treated the same way as your medical records are. Nobody has access to your medical records besides you and the doctor in charge. It should be treated at the same way, and we are treating it at the same way. We have, as I said, review boards and several regulatory bodies established already in place. So if an AI is, we probably won't use a ChatGPT version of it, but there might be a ChatGPT medical version of it. And it may be ChatGPT, it may be Claude, it might be Perplexity. There's about 100 of these out there, AIs. But a medical AI related with HIPAA regulations, at least in the US, and federal regulations added to it so that it is kept at the same level as your medical records should be. Some of these laws are already in place for us organ engineers and doctors. Some of the laws will need to be modified and adapted as AI grows and with the speed it's growing. So it's up to the policy advisors, but my suggestion would be to keep it as safe as your medical records are.
I see. Well, thanks for tuning in today. And if you stay curious and keep exploring the worlds of organoids, and if you have any question, leave us a comment below or DM us, and we'll see you in the next episode.
And with next episode, we're going to be talking about, now we're taking every disease that we work on and talk about it and how organ engineering can help us. So the next time, tune in to learn about autism spectrum disorder or autism-related disorders and their role, and how are we studying those using organ engineering.